So for our proposed experiment, we wanted to study the effects of biological sex on bean beetles and their metabolic rate. And at the beginning of the experiment, we came up with the proposed research question. On the screen, you can see, does biological sex have a significant effect on the whole body metabolic rate of bean beetles? And this is the oral presentation and oral lab report that we've prepared for you today on our proposed topic, and it's by Anika and Alyssa O.D. At the beginning of the experiment, we performed a literature search to kind of gauge what kind of information and what research had been previously conducted in this area, and we couldn't find a whole lot about bean beetles specifically and the effects of sex on the metabolic rate of bean beetles. However, we did find lots of information about similar experiments and studies being conducted on seed beetles and ground beetles. And this kind of highlighted a gap in the previous research that was done on this research topic, this specific research topic with bean beetles. Some of the information that we found was quite interesting. We found that the resting metabolic rate is positively correlated with the body size of beetles. And it trended with higher resting metabolic rates in male beetles versus female beetles. And this trend can allow male beetles' metabolic rate to reach up to 50% higher than female beetles. And then with age, male beetles' metabolic rate does decrease a lot more rapidly than female beetles' metabolic rate decreases. And then regarding the active metabolic rate for beetles, males and females' active metabolic rate is dependent on behavior and body mass. However, females' resting metabolic rate is not at all dependent on the body mass or behavior of the beetle. And with that, we found that female active metabolic rate correlates positively with the body temperature of the beetle, whereas males correlates positively with the body mass and size of the beetle. And the larger the body mass of a male beetle, the higher the metabolic rate will be, and the larger the body mass of a female beetle, the lower the metabolic rate of the beetle will be.
Based on the previous literature we found, we formulated our hypothesis, which was that the male bean beetles will have a higher whole body metabolic rate than the female bean beetles will. And this was under the same temperature conditions at 26 degrees Celsius and with the same type and amount of food as beans as a food source, which is our pinto or black-eyed peas. And our reasoning kind of behind this hypothesis that we formulated was that because of the previous literature that we did find and the studies done on other various species of beetles, they all suggested and showed that the male bean beetles in these studies generally all had higher body metabolic rate than the female beetles did. And so because of this, we thought that this trend would continue and keep consistent throughout our trials with the bean beetles, even though the bean beetles were a different type of beetle than the seed beetles and the ground beetles that were studied in the previous literature before. For our methods and materials, we started with step 1, and we separated our beetles by sex into 6 containers for each trial. There was 3 containers filled with 25 female bean beetles, and the other 3 were filled with 25 male bean beetles. And then for step 2, we labeled each trial container, for example, trial 1 male bean beetles, and then we did that for all three male bean beetle containers, and then all 3 female bean beetle containers. And then in order to set up the control group, like seen in the picture here, we decided to add 10 male bean beetles and 10 female bean beetles to a container and label it as such as the control group. And the control group was really important because it allowed us to compare the other trials data against the control group to see the differences in the metabolic rate with each sex singled out.
And then with step 4, we added 15 grams of black-eyed peas to each contrial, and added it to each container so that the beetles had sustenance for the 7-day period while we were not in the lab. And then step 5, we placed all 7 containers, so the 3 male trials and the 3 female trials, as well as the control group, into the 26-degree-Celsius fridge for the week. And then for step 6, we used the CO2 probe, and the software connected to the probe provided by the school, to measure the production differences between the sex treatment groups after the 7 days. And after gathering that data obtained by the probe and connected software, we analyzed the data to ultimately determine the CO2 production of each trial, the overall production levels of each treatment group and the control group, and as well as the metabolic rate of each treatment group and the control group. And then with all of that data, we performed some t-tests to determine if there were any significant statistical differences and how significant those statistical differences were between the 2 treatment groups we were comparing, as well as being able to measure them up against the control group that we had. This is our bar graph that we created at the end of the t-test that we conducted, and this is a bar graph of the average metabolic rate, comparing the difference in the female bean beetle metabolic rate and the male bean beetle metabolic rate. And as you can see, there is no significant statistical difference between the 2 sex treatment groups, because the p-value ended up being greater than 0.05. With the female metabolic rate ended up being a 0.278 parts per million CO2 per second versus the male metabolic rate, which was 0.129 parts per million CO2 per second.
So for the control group graph, this is what we created. And as you can see, the control group graph has slight inclines and declines, but the overall trend is an upward slope, which means that the metabolic rate of both of the sexes in the same control group container had an increase throughout the whole experiment. And the carbon dioxide production reached about an average of 751 ppm over the 600 seconds of the data recording throughout the whole trial.
So these are the 3 scatter plot graphs that we created with the female trial data. And the female trial graphs show a generally consistent increase as the time goes on, over the 10-minute period that the probe is in the Nalgene bottle. And this really does mimic the control group's overall trend line of a gradual increase over the time. However, the female trials have little to no decreases, whereas the control group has a slightly more dramatic decrease every once in a while. And the female trials are almost completely opposite to the males in terms of their trends, because the female trials are pretty consistent or are very consistent over the time. However, the male trials are pretty inconsistent, as you'll see in the next slide.
These are our male trial group graphs for each trials 1, 2, and 3. And as you can see, these graphs have big inclines and big declines, and less of a steady rate of incline of CO2 production for the male bean beetles. And in trials 2 and 3, we found that there was a positive increase in CO2 production overall, despite all the inclines and declines. But we found that trial 1, shown in the graph, did not have a positive overall production. And as you can see, the slope of the line of best fit was negative. And this just means that the CO2 production of the male bean beetles did not increase over the whole production or the whole trial period of the 10 minutes of measurement.
So in terms of the discussion and conclusion, we put together a few points that really were highlighted from the results. The metabolic rate of the female bean beetles was higher than those of the male bean beetles, by about 0.15 parts per million CO2 per second, because the female metabolic rate, as I said before, is about 0.278 parts per per million CO2 per second, whereas the male metabolic rate was at a 0.129 ppm CO2 over seconds. And the female group was even higher than the control group, and the male group was lower than the control group, and the control group was at a 0.0174 parts per million CO2 per second metabolic rate. And this does disprove our original hypothesis. And our original hypothesis was the male bean beetles would likely have a higher metabolic rate than the female bean beetles.
And we also found that the data gathered from the female trials, as seen in the graphs, was much more resemblant of the control group trials data in the graph that we created, because the slope of the line and the female trials in the bean beetles had a positive correlation with the amount of CO2 production and metabolic rate. Whereas the male trials were, since they were more up and down in steadiness, they did not mimic the control group as much. And obviously this leads to some inconsistency in the male trials as well. But overall, we did find that there was no significant statistical difference between the male and female trials. But even though this was true, the female bean beetles were, since they had a higher metabolic rate in the end, because of the higher CO2 production, the female bean beetles were more efficient in converting the nutrients in the beans in their food source to their energy that they can use. And because of this, their higher metabolic rate also means that they will have a higher energy budget to spend than the male bean beetles will, and they can spend this this additional energy on growth and reproduction after their maintenance and survival growth have been sustained. And because of the male trials and their increases and decreases, they don't have as much energy than the female bean beetles do.
So some possible points of error that we came up with after the end of the experiment and after all of the data and probe testing that we did, we found that the sex misidentification could have definitely been a possible source of error for the experiment, because for example, if there are male bean beetles in the female trials, then that could bring the CO2 production levels down of the female trials, and that would cause the overall average of the female treatment group for that overall average metabolic rate to be lowered, and this would jeopardize the integrity of the data we collected. And it is a pretty likely possibility because the process of separating the beetles between the 6 trials and the control group and making sure that the 3 male trials were getting only male beetles and the 3 female trials were only getting female beetles was pretty tedious and precise, and there is a possibility that we could have made a mistake. Another possible source of error for this experiment was the possibility that we miscounted the number of beetles that went into each trial. And similar to the possible sex misidentification source of error, the process of separating all of the 150 beetles into the 6 different trials and the control group was super tedious. So there is a very real possibility that a miscount could have occurred in one or multiple trials, or even in the control group. And this would be a source of error for the experiment because it would skew the data a bit. And if not every trial is getting the exact same number of beetles, then the CO2 production levels could be higher for trials with more beetles versus it would be lower for trials with less beetles. And in an attempt to combat this, we accounted for this predicted source of error in our experiment by adding a few more beetles to every container, so that if there was a miscount or any possible death occurred over the 7-day period, which will be explained later, it would hopefully attempt to eliminate this discrepancy and possibly help to eliminate the skew of data that this could cause. And despite our attempts, there is a possibility that a miscount still could have occurred, And this would ultimately skew the CO2 production levels, and all that data that we gathered using the CO2 probe, and the connected software. And ultimately, this would skew the metabolic rate, the average metabolic rate between the different sex treatment groups and the control group.
Our third possible source of error that we explained was some possible deaths between the trials. And because of the lifespan of the beetles, they're not very long. There is a possibility that some deaths could have occurred over the 7-day period in the bean beetles in each trial, while in the 26°C fridge between labs. But similar to the possible miscount of the number of beetles in each trial, the possibility of these deaths that could have occurred would skew the CO2 production levels of the beetles, because if the trials didn't have the same number of beetles we accounted for, or isn't experiencing the same number of deaths as each trial to keep it consistent, then the trials wouldn't be producing the same amount of carbon dioxide as each other. And so in turn, the skewed carbon dioxide levels would mess up the metabolic rate of the trials, and that would mess with the results of the experimental study altogether. Areas of future direction that we came up with could be researching how the metabolic rate of bean beetles is affected by the type of light they're exposed to in their environment. This could be seen in UV light or blue light or other types of light. And although this is not discoverable in a natural environment, because you don't find these types of light in the natural environment, because these bean beetles are oftentimes only used in a lab setting, this could help with this and determining what environmental factors and settings the bean beetles have the highest metabolic rate in to thrive in their experimental settings and labs. And our rationale for the future direction of this experiment was inspired by the labs we have done earlier this semester, like the Roma tomato plant lab, and we wanted to combine these two labs and the light intensity element that we studied in both of these labs, with some other sources, with what we did in this bean beetle lab. And so we thought it would be interesting to study the effects of different types of light, such as UV light or blue light, on the metabolic rate of the bean beetles that we have in this experiment. And these are references that we used to create our lit review, and the first two studies are done on different bean beetles, seed beetles and ground beetles.